Good afternoon, guys. In this opportunity, I will be presenting the guidelines for reporting the N-of-1 trials as it was reported by Boda et al. In the Council Extension for Reporting N-of-1 Trials 2015 statement. And essentially, N-of-1 trials are an important type of design that basically assesses the effectiveness of different treatments on one single patient. It uses evidence-based investigation, so a treatment decision is made for only one single patient. And if this type of design is effective or convenient in cases in which large trials are not able to respond to hypotheses or questions in the case of rare diseases, comorbid conditions, or in patients using concurrent therapies. And the idea is that these patients are so heterogeneous or so not special, not comparable to the population that in which this convenience studying them alone rather than in a group of in a randomized clinical trial. It is important that we review and learn how to properly report N-of-1 trials so these trials can then be replicated successfully and that they are valid. And studies have actually found that these trials are not reported properly. In many cases, they don't list exactly which is the primary outcome, and sometimes they also don't list the adverse events effects. In this table, the addition that were made in 2015 to the concert checklist are on the right. And if any line pretty much states empty, it means that the concert of 2010 was kept valid. So, for example, in the reporting of N-of-1 trials in the title or abstract as of December 2015, it is important that we identify the title N-of-1 trial, or if it's a series of N-of-1 trial that we list with title a series of N-of-1 trials, so the title right away can identify the type of design that it is. In the background and the objective, the scientific background explanation of rationale was kept, as of, required for the 2010 concert. But then it is now also required that there is a rationale provided for using the N-of-1 trial approach, like particularly, why are we using an N-of-1 trial in this case? And that we also list the objectives and hypotheses specific to this trial. Then in the method section, in the trial design section, we have to now describe the trial design, plan number of periods and duration of each period, including the running and washout is applicable, in addition series and whether or how the design was individualized for each participant, and explain the series design. So you know, guys, that it is important the washout period so we can make sure that there is no carryover effects from the medication. That's why it's important that now this is detailed. And it is important to that the changes to methods after trial start, like if there was any, for any reason, the eligibility criteria changed in the case of serious N-of-1 trials, or if anything changes, also important that we list that. In regards to the participants, the diagnosis or disorder, like the diagnostic criteria, the comorbid conditions, and concurrent therapies needs to be detailed, and also the settings and location where the data were collected, whether the trial represents a research method and reporting study, and if so, whether institutional ethics approval was obtained. As you can see, it is very important that we detail all the necessary information about why was this trial conducted, how, when, and all the reasons why we decided to run an N-of-1 trial approach on this patient. In regards to the intervention for each period, with sufficient details to allow replication, including how and when they were actually administered. And the outcomes, they kept with the completely defined pre-specified primary and secondary outcome measures, including how and when they were assessed. Description and measurement of property, validity, and reliability of outcome assessment tools, and any changes to trial outcomes after the trial commenced with the reasons why there were these changes to the trial outcome. In regards to the sample size, there weren't any changes made. We have to detail how sample size was determined and when applicable, explanation of any interim analysis and staffing guidelines. Remember that it is important that the interim analyses are planned a priori in the protocol of the study when it was in the preparation and the planning of the study. In regards to the randomization, there were few changes made. So now, as of December 2015, whether the order of treatment periods was randomized with rationale and methods used to generate allocations because it's important to be reported. And also, when applicable, the type of randomization details of any restrictions such as pairs or blocking. And this is more related to when we're doing a series of N-of-1 trials. Because if we're doing N-of-1 trials in only one patient, we can just pretty much describe pretty much the treatments and how it's randomized. Like say if we're doing a sequence of A, B, 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 A, we need to detail how was this decided and the fact that this was randomized. And if we're doing a series, the same thing. The allocation and concealment mechanism has to be detailed. This didn't change much. From the concert of 2010, the mechanism used to implement the random allocation sequence of sequentially numbered containers, describing any step taken to conceal the sequence until intervention were assigned, who generated the random allocation sequence, if done, who was blinded at the assignment, and a relevant description of the similarity of the interventions. The statistical methods did change. The methods used to summarize data and compare interventions for primary secondary counts must be detailed. And then for series, if it was done, methods of quantitative synthesis of individual trial data, including subgroup analysis, adjust analysis, and how the heterogeneity between participants was assessed, needs to be detailed as well. The statistical methods used to account for carryover effects, period effects, and inter-subject correlation. As you already know, it is very important that we take particular steps to account for the carryover effects and make sure that washout periods are sufficient before the change in treatment. Lastly, for the results, it is important that we list absolutely all the changes that were made, even if the protocol was shared, or even if we kept with the protocol. It's important that we detail everything, the number and sequence of periods completed, and any changes from the original plan with reasons. For this series, the number of participants who were enrolled, assigned to interventions, and analyzed for the primary outcomes. For series, losses or exclusions of participants after treatment assignment with reasons, and periods in which this occurred if applicable. And then in regards to the recruitment, whether any periods were stopped earlier and or whether the trial was stopped earlier. The days defined the periods of recruitment and follow-up stayed the same, and also a table that we put a baseline data table showing baseline demographic and clinical characteristics of each group. And then in regards to the outcomes and estimation, it's important that each primary and secondary outcome is defined and that any accompanying figure is displayed. For each primary and secondary outcome, the estimated effect size and its precision, and the binary outcome presentation of both absolute relative effect size is recommended. The ancillary analyses are also important to be reported. And in regards to the discussion, there were no changes made. We have to list the limitations, the generalability of the results, and the interpretation of the results. And other additional information is the registration and the number and name of trial registry, the protocol, where the full trial protocol can be accessed so we can see what was planned a priori and any sources of funding that was provided for the study. This is all for today, guys. Thank you very much and have a great day.